We're going to go through your experimental booklet. Now, this booklet is important because it summarizes and rather it consolidates all the different types of possible questions that will come up with regards to experiments. Okay, so how do you use this booklet? Now, the booklet is designed in such a way to help you see to give you a framework for answering different types of questions. So you go and memorize the framework. And then after that, you've got practice, you have practice questions whereby you can actually practice on your own, okay, to allow you to use the framework that was given to you earlier on. I will explain to you in a while. So first things first, okay, if you look under the learning objectives, you would understand that there are different types of questions. So these are the different types of questions I'm talking about. Are they asking you for aim? Are they talking about reliability? Are they talking about relationship? Are they talking about conclusion, fair test, control setup, hypothesis generating possibility, or accuracy based type of question related to experiments? So with each type of questions, you have a specific way of answering, and this is what this booklet will teach you to do. So for example, the first one, let's move on. Okay, so the aim of experiment. Now, the question that it will look like will be, what is the aim of the experiment? So the framework to use is to find out if change variable affects the measured variable. So what is the change variable? You will need to fill this in based on the question. Same with the measured variable. So example, the first question. Mr. Ko placed 5 identical fruits, A, B, C, D, and E in rooms of different temperatures and measured the time taken for each fruit to split. So right off the top, different temperature, okay, is your change variable, and the time taken for each fruit is your measured variable. Okay, then the question asks, state the aim of the experiment. So remember, use the framework given to find out, or start your answer with. So to find out how the temperature of the surroundings. Why? Because temperature of surrounding is actually your change variable. Affects the time taken for the fruit to split over here. Your measured variable. Okay, so this is a very systematic way of answering aim questions related to aim of experiment. Okay, now question 2, another example. Okay, Taryn set up the following experiment. She placed a light sensor at different distances. So your different distances in this case would be your change variable, and your measured variable is amount of light detected. So once again, to find out how the change variable, which is actually distance between torch and light, affects the amount of light detected by light sensor. Okay? Now, reliable results. Okay? Now, reliable results. Why do we want to repeat an experiment? The whole idea is you want the results to be repeatable. Why? Okay, you think about it. If you find something out, you want to make sure that the rest of the world is actually able to repeat your experiment and get a similar results. So you imagine if you are a scientist that invented a cure for COVID, right, through experimental techniques. You want to make sure that this piece of information or your results is replicable, it can be replicated throughout the world. Okay? So so these are the type of questions that they will ask you the way they will phrase it. How do we reduce experimental error? How do we ensure results are reliable? What can Peter do to ensure results are reliable? So he can repeat the experiment at least. Okay, at least. You can do more number of times. In fact, the more number of times, okay, the better, the more reliable it is. Okay, and then take the average results. So for relationship questions, what you want to do is the first thing is you want to identify what is the change variable and the measured variable, something which I've already taught you. Okay, and then you want to observe how the change variable affects the measured variable. So what happens? If change variable increases, what happens to the measured variable? Does it increase or decrease? Okay, vice versa. Okay, so therefore, this is the framework on how you can craft your answer. So if the change variable increases, the measured variable increases. Or if the change variable decreases, the measured variable increases. So basically, this is a framework of how you can actually craft your relationship questions. So let's look at an example. Naila and her classmate conducted an experiment using seed Y as shown below. Seed Y has a 7 cm wing-like structure. She dropped seed Y from the 4th-story corridor of a school building, while her classmate on the 1st floor measured the time taken for the seed to reach the ground. Naila and her classmate repeated the experiment 3 more times, each time trimming the wing-like structure by a little. First to 4 cm, then to 2 cm, finally no wing-like structure. Okay, then they tell you the results. So basically, first things to identify what is the change variable, like what I've done. The second thing is to identify measured variables. And what you can do for yourself is draw an arrow so that you know which direction is it heading towards. So as you can see, when I read it from left to right, I know that it's decreasing. So I write the word decreasing. Similarly, for my measured variable, I will do the same. Draw an arrow to show how is it being read, and then after that write down, is it increasing or decreasing. So one look at this, I know that when my change variable decreases, my measured variable will also decrease because they are moving in the same direction. So how do you then formulate your answers? It is to find out how the change variable, which is the length of the wing-like structure, affects the time taken for seed Y to reach the ground. Okay, so for the rest of the type of question for this particular relationship question, I'm just going to quickly scroll down and pause for you to try on your own, and then you can copy the do your corrections if needed, okay, for the rest of the questions. Okay, so I'm just going to scroll down for you to see. Okay, so many of these questions you will note that you've actually seen them before. It's just that we consolidated everything here for you. Okay, now for this question 10, I just want to pause a while to actually talk a little bit about it. Now, for this particular question, right, you do need to take note that, okay, there are 2 different types of variable here, okay? Now, one is talking about percentage of oxygen and one is talking about a number of red blood cells. So what you need to do is you need to find out what is the commonality between the two. And you will realize that the commonality is talking about increasing sea level. So because of that, okay, you need to see how does it affect. So there are actually two trends. You need to relate the two together. So in the first one over here, you realize that as sea level increases, okay, what happens is that your percentage of oxygen, okay, will decrease. And then after that, for the other one, as your sea level increases, your number of red blood cells actually increases. So write something like this to help you understand and then relate the two together. Okay? So then they ask you, based on the graph above, what is the relationship between percentage of oxygen in the air and the number of red blood cells found in the human beings with increasing height above sea level? Okay? So you need to say that as the height above sea level increases, because there are two trends, so you have to talk about two relationships. The percentage of oxygen will decrease, but the number of red blood cells will increase. Okay? Alright? Okay, and also please take note, relationship can have no effect also. Don't always think that it must be increasing, decreasing. It can be no effect also. Alright? Alright, moving on to conclusion questions. Okay, so this type of conclusions, basically you need to identify what is your change variable and your measured variable. And then you need to observe how your change variable actually affects your measured variable in a given data. So, in an example as shown below, Andy conducted the experiment shown below to find out how plants affect the temperature of its surrounding. Okay? So basically, with just this alone, I'm able to read that your plants is actually your change variable and your measured variable is your temperature of your surrounding. Okay? So, let's continue reading. He poured an equal amount of water into both pots and placed both setups under the sun in the field. Any excess water in the setups will flow through the openings into the beakers. Andy recorded the temperatures inside the beakers at the start and at the end of the experiment. The results are shown, okay, in the table below. So, from here, you can see the temperature change, right? So this is a negative, what? 33 minus 25, 8 degrees Celsius. Whereas this is basically a change of only 3 degrees Celsius. So setup A is the one with the plant and setup B is the one with no plant. So in your actual exam, you can actually use annotate like what I show you to make better sense. Okay? So basically, as you can see, setup A with the plant is actually able to lower your temperature more than setup B which has no plant. So then they ask, based on the results in the table, what can Andy conclude about how plants affect the temperature? So first step, identify the change variable, which is the presence or absence of plants. And the measured variable is the temperature, which we have already talked about. Okay, so what is the trend? You realize that with the presence of plants, it decreases the temperature of air, as shown in your experiment. Okay, so question 14 is another question that talks about conclusion. Okay, So once again, the question asked, okay, what can Carey conclude about the experiment? So remember step 1, identify the change variable. So how do you identify? Read the question. So he conducted an experiment on photosynthesis using a setup. He measured the amount of gas collected. Okay, so gas collected over here, you need to understand is actually talking about oxygen. And oxygen is due to photosynthesis. So when there is a higher rate of oxygen production, you will know that there is a higher rate of photosynthesis. Okay, so he repeated his experiment by increasing the number of tracing papers. So you know that because he increased, there is a change here. So this is your change variable. And what did he measure? He measured the amount of gas collected. So with that, they asked you, what can he conclude about the experiment? So if you take a look at it, as you increase the number of tracing paper, the amount of gas is actually decreasing. So you can actually write a short note. Okay, so as tracing paper increases equals to decrease in gas. Something simple like that to help you make sense of what is happening. Okay? So basically, you can then craft your answer by saying as the change variable, which is the number of tracing paper, increases, okay, the measured variable increases or decreases. So in this case, it's talking about your gas. Okay? Alright? Now, you also need to talk about what does your measured variable represent. Sometimes it's not so straightforward, okay? Because it's a conclusion, right? You need to ask yourself, amount of gas collected, like what I told you, represents your oxygen. And since oxygen is produced from the process of photosynthesis, we know that more oxygen equals to more photosynthesis. And that's how you're able to relate it. So basically, in conclusion, he can conclude that as the amount of light trapped by the plant increases, the rate of photosynthesis increases also. Alright? Okay, so this is another question on conclusion that you can attempt on your own. So as you can see, there's also another question on reducing experimental error. This got to do with reliability. So repeat experiment 3 times and take the average results. Okay, very straightforward. Same thing. Okay, please attempt this on your own and pen down the answers. Okay, so here are some common change variables, okay, and what they represent. So for example, okay, distance between submerged plant and light source is actually the amount of light. Now there's another word for amount of light that is of a higher level. It's called light intensity. In fact, when you reach secondary school, you cannot say amount of light or concentration of light. It's wrong. You have to use the word light intensity. Okay? Then after that, size of wing light structure, exposed surface area, okay, with the surrounding. Number of batteries in the circuit, amount of electric current flowing through the circuit. Okay? And then these are your measured variables. What do they represent? Number of air bubbles means rate of photosynthesis. Time taken for fruit to reach the ground, amount of air resistance. Okay? Number of paper clips, strength of electromagnet. What do we mean by that? So for example, you have a magnet over here. The number of paper clips that is represented talks about the strength. So this only makes sense if, let's just say, you have 2 magnets that are exactly the same size, okay, and everything else, and you got the same paper clips. Then let's just say this is A and this is B. So I can say that A has more paper clips attracted to it as compared to B. This shows or this suggests that A has a stronger magnetic force, okay, or magnetic strength than B. Okay, moving on to fair test. Now, in a fair test experiment, okay, they are concerned with your change variable. And you need to understand in any experiment, there can only be one change variable. Okay? So, how do they always ask you? So one common way of asking could be, is the experiment a fair test? They may ask you. Or do you agree that the experiment is fair? And so you would follow this framework if, let's just say, you think that the answer is yes. So first you say yes. In a fair test, there is only one change variable, which is this blank here refers to the change variable that is in your question. Okay, so you have to fill in a blank according to the question given, while keeping other variables like, okay, at least 2. Name 2 examples or variables, which I will explain to you. So if you want to say no, you don't agree that it's a fair test, this is how you can answer it. No, in a fair test, there should only be one change variable. Same thing, identify what is it supposed to be, while keeping the other variables, okay, what and what. However, in this experiment, which is also changed. So the second experiment in this case talks about, okay, the second variable that was changed, which is not supposed to be the case because there should only be one change variable, which I will exemplify. So let's take a look at this question. Clarice carried out an experiment to find out which material reflects light best. She prepared the setup as shown below using A, B, C. Explain why Clarice conducted her experiment in the cardboard box. So in this case, it is actually a fair test question. Alright? So in fair test, you're supposed to have only one change variable, which is your material. So basically, identify. So my material is basically my change variable. The rest of the variables needs to be kept constant. What are some of the variables? Let's talk about it. You have your lighted torch, which gives you your amount of light, your light intensity. Okay? You have your light sensor, it should be the same model. Okay? You should be in the same environment, even temperature, even everything. Okay? Now, if you do not conduct it in a cardboard box, there could be other light sources from elsewhere in the room, right? Or if you conduct it in an outdoor area, you can have natural light coming from the sun. So because of that, it becomes an unfair test, because your light sensor could detect light not just reflected from your lighted torch, but also from elsewhere. So remember, it is to ensure that the measured variable, okay, which is the amount of light detected by light sensor, is only due to the change variable, which is your material, and not other variables, okay, like surrounding light. So you need to tell me what is the example of the second change variable. So in this case, will be your surrounding light. Okay, now I know that earlier on I mentioned at least, try and give at least 2. But in this case, it's pretty specific that they are trying to prevent the amount of light being a second change variable. So you straight away talk about it. Okay? Moving on, next one. Gopal wanted to find out how material X affects the way cold water gains heat. The diagrams below shows 2 setups he used for his experiment conducted in the science lab. Okay, so remember, whenever you have 2 diagrams, I always tell you this, you need to compare. So the difference between this and this would be one has pocket X with many air pockets. So already from here, you know that air pockets is a poor conductor of heat, right? So you know that this one over here would actually gain heat from the surrounding at a lower speed. Okay, because it's only at 10 degrees, right? Normally, room temperature is about 25 thereabout. So heat should enter, okay, bringing up the temperature. Now, is this a fair test? Explain your answer. Okay, now the first thing I told you all to do is to identify what is your change variable, right? So your change variable is actually material X, right? So it's either it's present or it's not present. And then your measured variable talks about, okay, the rate at which it gains heat. So they ask you, is this a fair test? Okay, I don't think it's a fair test. Why? Because besides it being, okay, have one having material X, another one not having material X, okay, your beaker is also larger with a greater volume. In fact, there are 3 variables. Okay, what are the 3 variables? Different types of materials, or presence of X and absence of X, that's one variable. That's your change variable, whatever you're supposed to change. And then after that, the second variable, which is supposed to be the same, is your large and small beaker. And then you have the volume of water. So 3 variables are being changed. Not correct. In any experiment, only one. So answer, no, it is not a fair test. There should only be one change variable. Then you need to tell me what is the change variable, which is the presence of X. Okay, while keeping other variables like size of beaker and amount of cold water the same. Remember, I told you try to at least give 2. Can you see this is 1 and this is 2? Examples of what are the variables that are supposed to be kept constant. Okay, however, in this experiment, the size of the beaker and the amount of cold water is different. So you have introduced an additional 2 change variables to this experiment, making it unfair. Alright? So please go and look at the structure on how you can actually answer this type of question. Very important. Okay? So basically suggest the changes to make it a fair test. So think of things that you can do. Now wherever they ask you, right, to suggest something, right, please, it needs to be an action. Okay? You cannot say make the volume of water to be the same. So make is a very how do I say? It's a very general, non-specific, very ambiguous term. Make. How do you do it? So in science, right, you need to be specific. So you can say change. Okay? Change a large beaker to a small beaker. Or you can say things like use 100 ml of cold water for both setups. So you see how specific the answer actually requires you to be. Alright? So these are all other questions on fair tests. Please take a look, attempt it yourself, and then do corrections if you must. Alright? Okay? More questions? Okay, so this one I want to go through because it's a bit more higher level. It's a bit more thinking. Not so straightforward that you can use the framework. The key idea of a fair test is always one change variable. However, you can have higher order questioning like this that requires you to think in a more higher level manner. Let's take a look at this question. Haziq used the setup below to find out how the type of bulbs affects the temperature of its surroundings. So based on the first part, I already know already. So type of bulb is actually your change variable, and temperature would be your measured variable, just based on the first line. Bulb R was lit up. He covered the box and recorded the temperature of the air in the box over a period of 10 minutes. Then he removed bulb R and left the box uncovered for 15 minutes before repeating the experiment with bulb S, then bulb T. The results are shown in the table. Okay? Give a reason why Haziq left the box uncovered for 15 minutes before repeating the experiment with the next bulb. So this is actually a fair test question. Now, think about it. Okay, remember the whole idea of actually conducting an experiment is to make sure the experiment is fair. So your starting point must be the same. So in this case, right, he left, so you think about it, okay, what are they measuring? They're measuring temperature. So you think about it, if I actually tested bulb R first, and then straight away, immediately after bulb R, I put in bulb S. Okay? My question about it is, are you sure that bulb S results is solely due to bulb S and not because of bulb R? Right? So you think about it, the 15 minutes is left there so that the box has time to reset to room temperature, so that the box and the room temperature is the same, right? For all bulbs. Then that ensures that it's a fair test because you eliminate another variable, which is starting temperature of the box, right? It needs to be constant. So if you read the answer, it is to ensure that the beginning temperature of the air in the box is the same at 25 degrees when the experiment is repeated for the next bulb for the experiment to be a fair test. You see, it's all the same. If not, it wouldn't be. Okay? If not, you think about it, bulb R after 30 minutes, after 10 minutes is 30. So if I straight away start, it will be at 30. Okay? Alright, this is also another very interesting question that came up quite recently, last year's paper. I edited it. So in this case, they are also looking for fair test. But instead of putting the word fair, just fair test there, they ask you, how does it allow for a fair test? Okay? So in this case, you have to try and understand. One way you can do it is, okay, arrangement of containers. Okay, so you need to actually take a look and maybe draw it out, the pathway, to help you visualize that the distance is actually the same. Okay, from the entrance. And make sure you're very clear about your answer. Alright? Okay, this is trend. I've talked about it. We've gone through this question. Okay, next. I want to spend a little bit more time on this because this is something that many students don't quite understand. Now, experimental and control setup. So a control setup is one that's no variables that are being tested. Okay, so in short, right, whenever you see they ask you for a control setup, your control setup, control stands for CV, change variable. In your control setup, what you need to do is remove away the change variable, and that will be your control setup. Okay? So, do you remember there was this question that we did past year PSLE paper? They asked you about 2 bananas. They said that, okay, ethane gas, this word ethane gas. Okay, sorry, I think it's A. Ethane gas is said to ripen bananas faster. Okay, so then they ask you to design 2 setup. Okay, they give you 2 boxes. You have your experiment and you have your control. So they ask you what should be in your experiment and what should be your control. So remember this, your change variable, control means change, right? I said to remove it. So your control, since the change variable is actually your ethane gas. Why? Because you want to find out if ethane gas is able to increase the ripening speed of bananas. So in your experiment setup, you should have 1 unripe banana. Same for your control. Same, everything else must be the same. But you want to find out if ethane gas actually helps with ripening, speeding up the ripening process. So you need to put in the ethane gas. Now the ethane gas in this case is actually a ripe banana. They tell you that ripe banana releases ethane gas. Okay? For your control, you remove away the ripened banana. It's gone. Because the ethane gas is the control, it is the change variable. So you remove it, and that's why you do not see it in your control setup. Don't have. And this is the answer. Your control setup is just the unripe banana with no ripe banana, because your ripe banana gives it ethane gas, which is your change variable. So you remove that, and then you have your control setup. Okay? Now, so what is the purpose of a control setup? Say setup A. So it acts as a control to compare and confirm that the measured variable, so whatever. So in this case, this example, I will say that the ethane gas, the measured variable is the rate of ripening of banana, right? Is confirm and is to compare and confirm that the ripening time of the bananas is due to the, what is the change variable? The change in amount of ethane gas, and not whatever it is. Okay, temperature, surface area, so fill in the blanks. So let's take a look at this as an example. Martha wants to investigate if the number of leaves on the plant affects the amount of water taken in. So first things first, I told you already, always talk about change variable and measured variable. So this is your change variable, and this is your measured variable, right? So Martha has 3 setup of similar plants and 3 500 ml glass beaker. She may use some or all of the items. She filled the 3 glasses with 400 ml of water. What items should she put inside for each? So very simple, I told you, right? Remove away the change variable. This is what we need to remove for control. So you can have experiment 1, experiment 2, and then after that you should have a control. So for control, you want to remove away change variable. So change variable in this case is actually number of leaves. So in short, you should have a plant that has no leaves on it. Remove away the leaves, right? And then you've got experiment 1 that has all the leaves. And then after experiment 2 is lesser number of leaves. Okay? So beaker 1 with all leaves, beaker 2 with fewer leaves, and control setup no leaves. Remove away the change variable. Okay? That's it. Very straightforward. Okay. We can try this again. Jin Long wanted to find out whether germinating seeds produce heat. he soaked some seeds in the water before placing them in a glass container. So first things first, always identify change variable. Okay? So your change variable in this case is whether your seeds are germinating or not. This is the change variable. CB. And then your measured variable is actually heat. Right? Now, then after that, suggest a control setup to show that the rise of temperature was caused by germinating seeds. So everything else should be the same except that your seeds cannot germinate. Right? There shouldn't be any germinating seeds. So in this case, everything else is the same, but you remove away the soaked seeds. Right? So in other words, in your experimental setup, you put back the soaked seeds. If there's a rise in temperature, it tells you that it's due to the presence of the soaked seed. Okay? There's the unripened and ripened banana. Okay, the one that I was talking about. There you go. Okay? So, I did talked about already. This one, I just want to stress one more time. You cannot exceed whatever amount they give you. So, in this case, 2 and 2. Okay, for both control and experimental setup, the maximum number you have is 2. Okay, so you cannot times 2. You cannot say, oh, experimental setup 2 unripe banana. Then control setup 2 unripe banana again. Unripe banana again. No, cannot. Okay? Now next, hypothesis. This is something that is not very, many of you are not very familiar with this. So, a hypothesis basically is a prediction of outcome based on scientific reasoning. So in the recent years, right, they may ask you to state a hypothesis. Okay, and then, basically is to state a prediction. So how do you phrase it? So for example, if my change variable increases, my measured variable should increase or decrease or remain the same. Okay, it's a little bit like relationship, but the difference is this. In a relationship, is it predicted? No. A relationship question is not a prediction question because it is proven by the graph given to you. Okay, so if you have a graph, okay, your relationship that you state is proven in the graph. So it is not a prediction question. So if the graph shows that increasing, if the graph shows like something like this, okay, they show you a graph. Okay, this is amount of light. This is number of bubbles. And it shows something like this. As amount of light increases, number of bubbles increases. This is not a prediction. Then they ask you, what is the relationship? This is something that a relationship that you see from the graph. So hypotheses don't have graph one, no data one. Okay, you have to predict based on your scientific reasoning. Okay, so for example, like what? So I would say so they will give you some background knowledge and then they ask you to come with a hypothesis. So something that I'm very familiar with, I would say, if light intensity increases, that's my change variable, rate of photosynthesis will increase. Why? Because due to my own scientific knowledge, I know that in order to photosynthesize, your plants needs light. So if I increase the amount of light, then my photosynthesis should also increase. This is based on my own scientific knowledge or the knowledge that was given in the question stem itself. Okay, or I can say if the change variable is missing or present, then the measured variable will increase or decrease or remain the same. Okay, so let's take a look. Mr. Koh placed 5 identical fruits A, B, C, D, and E in rooms of different temperatures and measured the time taken for each fruit to split. The results of his experiments are shown. So, in this case, right, he gives you like a data. Okay, so based on this, you should be able to predict. But this is not for sure, you know, not certain one. Okay, but you can predict. So remember, identify what is the change variable. So in this case, different temperature is the change variable, and your measured variable is the time taken for the fruit to split. Okay, so remember, I can use this. So looking at it, what can I so-called predict? So if I take a look at it, as the temperature increases, I notice that, hey, the time taken actually decreases. Right? So basically, I can say that my change variable is different temperature, right? So as, okay, or if the temperature of the room increases, the time taken for each fruit to split, okay, should decrease. Okay? Phrase like this. The word if needs to be there for hypothesis. This is different from relationship question. I want to point it out. The word if, because it's a prediction. Okay? And this is what scientists do. So, like for example, right, when I'm a scientist, right, I conduct experiment to prove whether the hypothesis I came up with is true or false. Right? So it's like something saying like, if I add chemical X to COVID-19 virus, it should die because prior to this, I know that based on some data, chemical X is able to eradicate SARS virus. So based on that, I come up with a hypothesis. But is it absolutely true? I don't know, because it's based on data. Okay, and then I'll need to conduct an experiment or more experiment to prove. Okay, let's see another one. Okay, Tareen set up the following experiment. She placed a light sensor at different distances from the torch as shown below. Okay, so once again, highlight what is your change variable, okay, and your measured variable. So this is change variable, this is measured variable. So state the hypothesis, okay, as or if distance x decreases, the number of light detected should increase. Okay, maybe we change this to if to make it more consistent. If distance x decreases, the amount of light detected should increase. Okay? Right, here's another one. Change this to if for more consistency. Alright? Because this is hypothesis, not relationship question. Okay, so this is another more not so straightforward hypothesis question. Okay, so let's take a look at it. Swimmers in the ocean wearing shiny jewelry are found to be bitten more by shark pee. A scientist wanted to find out if shark pee are attracted to shiny objects. He used the setup below with one shark pee in a pool. It is noted that shark pee is dark grey in color. Okay, the scientist counted the number of times shark pee touched the ball for 12 hours. He repeated the experiment using another ball with a dull surface. Okay, he then recorded his results as shown. Suggests a hypothesis on how jewelry will affect its attractiveness to shark pee. So because it has a shiny surface, right? Okay, it actually attract them more. So maybe we can modify this answer to make it look a bit more like the one that we are familiar with right at the start. So remember, there's two ways of answering. One was talking about if, right? The other one talks about absence or present. So in this case is absent or presence of what? Shiny surface, correct? So if what? If the jewelry has shiny surface, the shark's pea would be more attracted to it. Right? Okay? Moving on, generating possibilities. Okay, this is something that is new. So generating possibilities talks about exploring other alternatives, new methods, possibilities, and beyond the obvious or preferred one. So let's take a look at this. Priya set up an experiment using a plant with only green areas. Okay, she wanted to find out whether presence of sunlight would affect the amount of starch produced in a plant. She placed the plant in a dark room for 2 days. After 2 days, she placed the plant in a well-lit room. Okay, she had the following materials. How could she use leaf X and Y and the materials to find out the presence of sunlight? To find out if the presence of sunlight affects the amount of sugar produced. So basically what they're asking you to do is generate possibility. How do you use whatever is given to you to come up with a possible method of testing? Alright? If presence of sunlight affect the amount of sugar produced. So basically you have to make use of what you have. So you know iodine solution is able to do a starch test. Okay? And then your black plastic bag will block out light, whereas your clear plastic bag will allow light to pass through. But you need both because it needs to be a fair test. So one for each leaf. Okay? So basically, let's take a look at the answer. She can cover leaf Y with the black plastic bag and leaf X with the clear plastic bag. Then conduct a starch test using iodine solution at the end of the experiment for both leaves. Right? So that is what you could have done. Okay, this is another interesting one. Cell X is an organism that has features of both animal and plant cell. Okay, so when cell X was viewed under the microscope, it was observed that part P had a green pigment which could be found in all parts of the plants. The eye spot of cell X could guide the plant cell to move towards a light source. Suggests how this ability enabled cell X to survive. So as you can see, this type of question is not something that you commonly see, but it can be related to what you have already known. So the fact that you know that it has chloroplast, which means that it can photosynthesize. And in photosynthesis, you need light. So if it has a eye spot that is able to spot light, okay, it will increase the amount of light for more photosynthesis to take place. Okay, be careful. In this part is more, because in a pond or wherever it is, the organism could be already receiving an X number of light. You want to increase this, okay, by going to the source of it. All right, so then you can look at this and then attempt this on your own. All right, so this is another question related to the shark. So then they ask you, suggest how the scientists could use his result to reduce the number of attacks on the swimmers by animal P. So basically, since, remember, it's generating possibility, right? So you generate a solution. So if you know that the shark is attracted to shinier objects, then what you want to do is, you want to advise, okay, the swimmers not to wear any jewelry or shiny items, right? Then you won't be, your shark P won't attack it, okay? Now, accuracy. So accuracy is talking about how your measured variable can be obtained precisely. Now, I've talked about this in very, very great detail during our normal lessons already. Okay, so you are concerned with the measured variable when you talk about accuracy. So, for example, the instrument used to obtain the measurement. So, for example, if you are measuring weight, definitely a normal weighing scale, okay, that has a decimal point of only 0.1, is not as accurate as a digital scale that has 0.01 accuracy. Okay, ruler is the same thing also. Okay, a meter rule is not as accurate as a vernier caliper. It's not in your syllabus, but the whole idea is a vernier caliper can measure up to higher accuracy. Okay, it's also about how your method of measurement, okay, is the best possible way to obtain the measurement that you want. What do I mean by that? Let's take a look. Okay, now this was basically a question that was very similar to what we've attempted. Okay, so John placed the same amount of seawater which contained living organisms into two identical glass containers A and B. Okay, he then placed A and B into a dark room, while B was placed under a brightly lit room. He proceeded to record the number of carbon dioxide in each container hourly for 6 hours. Explain why the amount of carbon dioxide in A increased while the amount of B decreased. Okay, this is straightforward. It's not talking about accuracy. This part suggests one possible reason that it could have resulted in John obtaining inaccurate results. This is talking about accuracy. So remember, he wanted to measure the measured variable, if you recall, was actually concentration of CO2. And he did this hourly, you know. So can you imagine every single time he opens the lid, some of the carbon dioxide will escape. So it's not very accurate, what? When you think about it, right? Okay, so how do you prevent that? You put in a carbon dioxide probe, okay, that can measure the concentration of carbon dioxide without opening the lid. Okay, this question, the sharp question, once again, is also this question was, just in case you didn't know, was adapted from your mosquito question. Okay, so I designed it in a way that I didn't want to plagiarize the PSLE, so I changed some things. Okay, so the question asked you, scientists use a pool that has white tile walls. Okay, how does it allow it to attain more accurate results? So because the measured variable in this case was actually number of times the shark touched the floating device. So you need to see it clearly. The shark head is gray in color. So you want a background that is contrasting to the shark, so you can see the shark clearly every time it touches. Okay? So you can count accurately. Alright? Okay, you can take a look at this. This tells you how this question talks about how you can use a data logger to have a better, more accurate measurements. Okay? Alright, so a normal thermometer may not be able to capture it so accurately due to human error, due to the fact that okay, the intervals of accuracy is not there. Okay, it's not, it only can read up to 0.1 accuracy. Okay? Alright. So with that, we've come to the end of this experimental techniques. What you need to do now is to look through the video and then really write down whatever you need to jot down, note down whatever to say. So this booklet is very useful for you, especially when crafting answers, okay, for the various type of questions